I wanted to give a brief refresher course for what we talked about last time, and also just an introduction for those that weren't able to be part of the last presentation. And then Galit's going to talk about the variants, and then Dan's going to talk about the Janssen vaccine and why that is going to make such a difference in the world. So just to move on, it was in 2019 in December, this new pneumonia appeared. Nobody knew what it was. And within 5 weeks, we knew what was causing it and we knew how to diagnose it, which is just phenomenal. When you compare that, for example, to HIV, the same process took about 5 years for HIV. And that's because of all the scientific progress that's been made. What the cause was this virus called SARS-CoV-2. It's a coronavirus, and it has genetic material in the center that basically takes over the human cells when it infects them and forces them to make new viruses. The way it gets into those cells is that it binds to a specific receptor that happens to be on cells in the lung and the nose, and that's why the first manifestation is pneumonia. It's because virus is replicating expressly in those cells, but it can then disseminate to other parts of the body. The body fights back by generating antibodies. You all have white cells in your body, and those white cells, when they encounter a virus, some of them will be inherently able to recognize a new pathogen like SARS-CoV-2 and generate these antibodies, which are little proteins. And what those antibodies do is they bind to the spike protein on SARS-CoV-2 and they prevent it from attaching to the receptor. So in an ideal world, you generate antibodies and that happens when you get exposed to the virus. Those things get generated and then they start to combat the virus, and then the virus goes away. And ideally, what happens is that as those antibodies are generated in an infected person, the virus is eliminated and you're left with these antibodies that give you protective immunity, ideally for your life, but it turns out that it's shorter than that for COVID, and I'll get into that here on this slide. Part of the problem with COVID disease is that the virus itself causes immune system damage, and I'm showing that here by this red line. So there's a lot of damage early on, even before the antibodies are made, and when the antibodies are made, because of that damage, you never get really great levels of antibodies, or you often don't. And also, those antibodies often don't persist like we would normally expect them to. And I think Galit will come back to say a little bit more about that. What we're trying to do with vaccines is actually prevent that damage from occurring and instead teach the body's immune system how to fight the virus without it ever seeing live virus. And the way we do that is by, for example, just making the spike protein, which antibodies recognize, and generating an immune response to them. So the idea here is that you either in the body with the mRNA vaccine or the adenovirus vaccine or other vaccines, you generate, you basically get this spike protein to be made in the body, and then antibodies are generated against it, and you're left then with antibodies that hopefully provide durable immunity. Now, ideally, with no immune system damage, because you've been immunized, now you have these antibodies around that persist, and hopefully at higher levels, and provide protection then if you ever get exposed to the virus. So that's essentially what we're trying to accomplish. Some of these vaccines require 2 doses, and the reason for that is that you often get a boost right away and then a bit of a decline, and then you need another boost or a second immunization to get up to really high levels. And that's the case for the Moderna and Pfizer vaccines, but it's not the case for the Janssen vaccine, as Dan will show you, which requires only a single dose. So that's a very brief introduction to vaccines and how they're generated and how we go about that. And that sets a stage for the second part of the formal presentation here from Galit, which is about these mutants that you've all been hearing about and whether we should be worried about those. So Galit, why don't you go ahead and just let me know and I can advance the slides.
Okay, thanks, Bruce. So hi, everyone. So I have to say that I was just at a conference today, a virtual conference on viruses, one of the largest conferences that Bruce and Dan and I normally go to, and we can't all see each other in real time, but we can see each other in virtual space and share data. And there was some incredibly optimistic data that was presented at the conference. And I just want to start off this part of the presentation with that. And the news out of South Africa is that although the variants are potentially evading some of the immune response that we've induced with antibodies, that the antibodies that are raised against the variants are still protective. So that was one of the best pieces of news that we heard. And I'll tell you why that's so important and why we know that is the case in the following slides. So if you go to the next slide, so as Bruce mentioned, what happens is following a virus infecting a cell, what ends up happening is that the virus will essentially replicate. It'll come out of the cell, it buds off the surface. And during this process, the virus has to basically remake its proteins, generate its own nucleic acids, and package itself all up so it can make more progeny that go on and basically keep that infection going. Now, if we go to the next slide, what is the major point is that during this process where the virus is going into the cell and making new copies of itself, errors are made in that virus. And those errors lead to little spelling mistakes, essentially, in the virus, as you can see here in the little blue dots. And these little spelling mistakes, if they're not picked up by the virus and they don't affect the viability of the virus, they essentially get packaged in. And this leads to the generation of new progeny, right? We all hope that our kids are going to be better than we are, in the same way that viruses evolve and progeny viruses will incorporate some diversity that can lead to the creation of these small changes in the actual virus. Now, those viruses may not be important in most situations. Those changes may not be important. They might just be mild, very benign changes. But once in a while, those changes can influence the way that our body sees the virus. And those are the mutations that we care about. So there are variants happening in SARS-CoV-2 all the time. Every single time the virus infects people, we will have some modification of these viruses. But the variants that we care about, we call the variants of concern, as many of you have maybe seen in the media. And those variants of concern are the ones that essentially end up dominating across populations, which means they begin to spread more effectively. Now we'll talk about why that happens. If we go to the next slide, the variants of concern that we care about are the variants that occur in the way that that allow the virus essentially infect more effectively, or that are able to escape our immune response. Now, what happens on the left side is if you have a variant that gets generated because there's a little mistake in the virus, but it's kind of embedded in the protein, it doesn't really make a difference, but it still allows the virus to interact with its host receptor called ACE2, then those changes may perpetuate, they may not, they're not selected, but they basically stay within the virus and they have innocuous effects. Now, sometimes a variant will occur in the middle, and that variant will occur in a place that the virus does not like. That variant will essentially abrogate the ability of the virus to interact, and those viruses will not get selected because they will not be able to infect, so they will not propagate or make new baby viruses. But on the right side, once in a while, you will have a modification that occurs in the virus, and that modification will actually allow the virus to bind better to its target receptor. And that is a variant that will gain access to more cells and will eventually potentially propagate more efficiently, even across populations. That is a variant of concern. Now, whether that variant is going to cause more disease is the big question. And what I can tell you so far is that we do not know if any of the variants that are currently the variants of concern actually cause more fatality. But what we know for most of these new variants of concern is they do spread more efficiently because they are selecting for adaptations in the virus that allow them to interact better with this host receptor. These are evolutionary changes that are totally expected in viruses. If this did not occur, we would be shocked as virologists. So that is the most important point that we always try to stress. This is an expected change. As the virus leaped from bats into humans, it is bound to adapt to the population. So that is the first kind of variant that we want to talk about. If we go to the next slide, the bigger issue, and this is what we're going to talk about today, particularly in the context of vaccines, is that what we know and what Bruce mentioned is that the way that we, as an immune response, interfere with infection, the types of immune responses we evolve are driven by antibodies. And these antibodies aim to basically bind to the SARS-CoV-2 spike and essentially block its ability to interact with the host receptor ACE2. These are the antibodies that we believe are critically important for preventing infection, both following infection as well as following vaccination. The critical problem is when a variant is able to also block antibody binding. And those are the kinds of variants that we are watching carefully to see if these are evolving and to make sure that these variants are not going to interfere with the vaccine-induced immune response. So if we go to the next slide, the idea here, right, is whether or not the antibodies that we're generating are going to be impacted by the variants of concern. And so this is really where the immunological community is working literally day and night since the identification of these variants of concerns to really begin to understand the impact of these variants that are emerging, to understand if they are truly just adaptations for spread versus if they are adaptations that are going to make our vaccines potentially less effective. So if we go to the next slide, the idea is that we, of course, make lots of antibodies to these particular viruses. And so the question is how do we make antibodies now that are able, through vaccination, to not only see one spike, but to potentially see all the different potential variants? And so what we're looking at here is the idea of what Bruce had shown us at the beginning, where we have come up in the vaccine development space with the idea that we can expose the immune system to just the spike antigens to drive these protective antibodies that will essentially block viral infection. And that's in the middle of the slide. But now, because we understand the variants that are popping up, we understand where these changes are occurring, we are coming up with novel vaccine development ideas and designs that will allow us to now potentially also boost or potentially deliver just these new spikes that are emerging in the population to drive immune responses that can see not just the original virus, but all these different variants. So essentially boosting the response to see any virus that might pop up in the population. So there's two different strategies that folks are exploring. Both we've heard in the news from Moderna and Pfizer is the idea of basically immunizing individuals with these variant species that are shown in the top in option 1, where they've basically changed the sequence to include these new modifications. The idea there is they might be able to boost the vaccine-induced immune response and broaden antibodies to see all these new variants. And in the bottom, there are folks who are coming up with even more interesting, or let's say sexier designs, where these designs now, instead of just going after trying to immunize and induce immunity to one variant, they're basically anticipating all the potential variants that are popping up in nature, including variants that are coming out of the UK, variants from Africa, variants from Brazil, and even other variants that are starting to appear in California and New York. And they're making designer cocktails of spike antigens that will essentially drive the immune response to basically see potentially any new evolving SARS-CoV-2. So if we go to the next slide, the idea is that using these new designer cocktails, we are going to have next generation SARS-CoV-2 vaccines. So we're already in an incredible place with the current cocktails of vaccines. So the mRNAs, the ad vectors, as well as the proteins that are emerging soon on the approval market. But the idea here is that using these next generation vaccines, we have a tool in our back pocket that will prevent any possible liabilities that might occur in the future with any new emerging variants. The idea here is that we can either deliver these new variant preventing vaccine cocktails to the immune system right up front, and then boost with these types of vaccine antigens and drive very diverse swarms of antibodies that can see all these viruses. Or alternatively, we can boost with the current vaccines that we have already in our back pocket, drive a protective response against these dominant viruses that are already circulating, and then come in and boost with these new variant approaches that will now spread the immune response and make it really give us that ultra high level of protection against this naturally selecting pathogen. If we go to the next slide, what we've seen, though, over the last few months is this explosion of vaccine technologies, right? This is an unprecedented moment in vaccine history. We have never seen the entire world refocus their attention to a single pathogen. And the result of that explosion of attention to this particular virus is the development of so many new vaccine platforms that are all targeted to trying to drive the most protective immunity against SARS-CoV-2. And so what I'm showing you here are probably the dominant, most exciting platforms that have emerged over the last year, including on the bottom left side, the genetic vaccines that include the mRNA vaccines that really are, brand new tools that are unbelievably flexible, incredibly rapid, that allow us to basically program the immune response through this novel technology. We also have DNA vaccines that are coming up in the back, right behind these mRNA vaccines. And DNA is very cheap, very stable, another really exciting tool that's coming up. We've seen inactivated viral vectors coming out of the East, really exciting technologies that are really born from Louis Pasteur 200 years ago. So ancient technologies that are really, those are the inactivated particles, on the right side. The inactivated viral vectors that come born from this man, Dan Barouch, who you might all know here on the call. The viral vectors essentially use innocuous viruses that cause common colds or really don't cause any disease. And we use these viral vectors as Trojan horses that basically bring in cargo the nuclear material or the genetic code for the antigens we want to train the immune system to see. And these viral vectors act as magnificent stimulators of the immune system. And so this is what makes these particular particles that are stable, easy to distribute globally, a remarkable tool that we're seeing realize and probably have the biggest global impact on turning the tide on this pandemic. We also have viral proteins that are older technologies that we have from more than 100 years that we've been using for flu vaccines. This is a little bit of slower technology, but a tried and true and tested technology that we have in our back pocket. And this is the basis of Novavax and Sanofi products that are coming up in the next few months through the clinical development pipeline. And finally, the inactivated SARS-CoV-2 on the right side, as I mentioned before, which was really the inception of Louis Pasteur, where we can take the actual SARS-CoV-2 particle, make sure it is no longer infectious, and induce immunity. And this is really a technology that is being used very actively in China. And there are many other technologies which we're really excited about. We can talk about more, but the one thing that I have to say, at least from the Ragon Institute perspective, is that this is the first time in history where we can study the immune responses induced by all of these vectors, all of these platforms. What that means is we can start to understand how we can selectively engineer the immune response. What the implications of that is that for the next pandemic or for HIV, we will now have a Chinese menu of potential options of platforms that we can essentially use to now drive immunity that will provide protection against our pathogen of interest. It's truly an unprecedented time in our history as vaccine developers. Okay, so if we go to the next slide, I'm going to pass it over to Dan.
Dan, oh, there you go.
Yeah. Okay, so I guess I'll tell you from more of a personal story perspective what we've done over the last year and where we see it going. So next slide. So this really picks up on Galit's slide, and now in the United States we have 3 approved vaccines. The 2 mRNA vaccines from Moderna and Pfizer, which were approved by the FDA at the end of December, and the J&J, the Janssen vaccine that we'll tell you more about, because that's the one that we helped develop. That was just approved about a week ago. And we're hoping that that's not the end. We're hoping there's actually going to be 2 more vaccines this spring. One from AstraZeneca, which also falls into the inactivated viral vector category, and Novavax, which is a purified protein vaccine. And that will really round out the 5 vaccines that are likely going to be deployed in this country. We don't yet know the results from the AstraZeneca or the Novavax studies in the US. That remains to be seen. But we know that there are at least, we know that in the United States, we will have at least 3 vaccines, all of which are now proven to be safe and effective, deployed all at a very large scale. I think you probably heard the news last week that Merck is going to partner with J&J for the production of the Janssen vaccine. And because of that, then there's going to be a vast increase in vaccine supply, a vast acceleration of the vaccine rollout in the United States and throughout the world. So much so that President Biden announced that there's going to be vaccine available for every person in our country by May. Without the Merck partnership, it would have been July. And if the Janssen vaccine had not been successful, then it would be sometime in the fall. So hopefully what we can do is we can essentially get the country and get the world immunized more quickly. Next slide. So this is what we did. This story, this project of developing a COVID-19 vaccine started on January 10th of last year. But actually, the project actually started 15 years ago with the initial development of the adenovirus vector platform system, which was developed essentially between 2003 and 2005. Not for COVID-19, of course, and not for any coronavirus, but actually it was part of our HIV research program and our HIV vaccine program. And I think I can say with confidence that if it wasn't for our HIV research program, there would be no J&J COVID vaccine based on Ad26 today, because that technology simply would not have existed. And the Ragon Institute was instrumental in helping to move the HIV vaccine platform based on Ad26 forward, both in animal models and clinical trials. And the phase 3 studies with the HIV vaccine are still in progress. So during the course of just a portion of the time required to do the HIV trial, the entire COVID vaccine program went from beginning to end. So we really leveraged our HIV research for the development of a COVID-19 vaccine. And it's clearly obvious, both from a technology point of view, from an assay point of view, from a collaboration point of view. And so I think it shows the power of science and the importance of different scientific fields feeding each other. I can say that I think the development of multiple COVID-19 vaccines in just a year is nothing short of a stunning scientific accomplishment throughout the world. And it goes well beyond the individual groups that develop those vaccines. It really is the product of decades of advances in science, in virology, in immunology, in vaccinology, in manufacturing. And what we're seeing now is the culmination and deployment and the focusing of advances in science from decades. If we look very specifically at what we did, on January 10th of last year is when the sequence was released of the novel coronavirus, as it was called then, which later became known as COVID-19. And we started working on the sequence immediately. We started working on the sequences over the weekend. That January 10th was a Friday evening. We started analyzing the sequences and developing electronic sequences over the weekend. So when we got back to lab on Monday, January 13th, then we were in a place where we could start ordering synthetic genes and actually begin the vaccine design and development process. Over the next few weeks, then I had a few very important conversations. One was with Galit. We were having lunch together, eating barbecue food. This was long before there was any social distancing whatsoever. And we were talking about the need to make a vaccine and how to actually develop it and how to show it works and what assays are going to be needed to test it. Second conversation was with Bruce Walker, who just got back from South Africa on a trip in early January. And I think giving me a call was one of the first phone calls Bruce made when he got back to the US and said, what do you think of this news coming out of China? That was somewhere in maybe the second week in January. And I said, I think that we're going to need a vaccine for this. And Bruce immediately said, yes, we do. And then there were conversations with Terry and Susan and Mark and Lisa Schwartz and others of how to actually start that and make it happen. And then I had another very important conversation with my colleagues from J&J towards the end of January. I called my colleague Johan van Hoof, who's the director of vaccines at Janssen, and we had worked with them intensely over the last 10 years in the development of our HIV vaccine candidates, as well as a Zika vaccine candidate, as well as other vaccine candidates. So we had a very deep relationship with J&J. Obviously, we would need a pharmaceutical company to actually manufacture and distribute this vaccine. We couldn't do that as an academic group. So at the end of January, very, very quickly, all of that materialized into a very rapid and a very serious vaccine development program that involved philanthropy, that involved academia, that involved government, that involved industry. And it really was focused on developing this vaccine as quickly as possible. So we started preclinical studies in animals to test different vaccine constructs in February. We also produced viral challenge stocks to do animal testing, which was done essentially in March and April. By the end of March, we jointly decided with Janssen the very specific vaccine constructs to go forward with. The first in human clinical trials were started in July. And based on the early clinical data, we started phase 3 efficacy trials in September. And as you may have heard in the news, the top line results were announced on January 29th. And most recently, a week and a half ago, on February 26th, was the FDA VRBPAC meeting where all of the efficacy data was presented to the public and then was approved by the FDA and the CDC about a week ago, I think 9 days ago in particular. And then over the last week has been the first week of the rollout of the J&J vaccine. 4 million doses have been distributed across the country and have now, most of them, I think, gone into people's arms. Over the course of this year, J&J now together with Merck have a plan for a massive scale up and distribution of the vaccine. The first 100 million doses are going to be delivered to the United States, but there's a commitment for at least 1 billion doses of this vaccine to be delivered to the world. And characteristics of this vaccine, including the single shot vaccine, as well as the lack of a need for sub-zero freezing, makes it a vaccine that really can reach all 4 corners of the planet, including the most remote areas in the developing world, which really could not be reached by some of the vaccines that do require either multiple doses and or sub-zero freezing. So we're incredibly excited, and we really hope that collectively all the vaccines can now be used to save lives. Next slide. So the Ad26 vaccine is constructed as Galit described. It's an attenuated common cold virus, and into the common cold virus, which we made so it can't replicate in humans, so it can't cause a disease in humans, but rather it's used to essentially package and to transport the coronavirus spike protein and to help transport it into cells. Once it delivers the COVID-19 spike protein DNA into cells, the protein is made, and then the spike protein induces antibodies as well as T-cell responses in the human host. It turns out that process is very efficient, so efficient that this is the first single shot vaccine that's been approved. Next slide. So the results of the phase 3 study were updated on February 26th, which was the FDA advisory meeting, which was a public meeting. So anyone who wanted to invest 8 hours of their day could have heard all the presentations. I think they're even available online. The key thing is that the phase 3 trial was conducted, it was a very large study that was conducted in 3 geographic regions, in the United States, in Latin America, including Brazil, and in South Africa. It was a very different study than the ones conducted last fall for the RNA vaccines. And that's, I think, very important to realize. The J&J study was conducted at the peak of the global surge, with the efficacy collection time period being December and January. Also, in regions of the world where there were the circulating resistant variants that we've all heard about, including the more contagious variants, as well as the resistant variants that have compromised the efficacy of other vaccines. In Brazil, over 70% of these circulating viruses were the resistant so-called P1 and P2 viruses. And in South Africa, over 95% of the circulating viruses was the resistant so-called B1351 variant. Despite these dramatic epidemiologic shifts that occurred, the protection against any symptomatic infection in the study was quite good, 72% in the US, 68% in Latin America, and 64% in South Africa. But the most important finding of the study is the protection against severe disease was 85%, and that was really unchanged from geographic region. And the most important endpoint of all is the protection against hospitalization and death, which was 100% globally. So no one who got the vaccine in any region of the world became hospitalized or died, which is quite a stunning result given the global surge in the presence of the resistant variants. We would never have expected a vaccine to perform this well. Next slide. So this is the actual data from the trial. This is prevention of severe disease. On the y-axis is the incidence of severe COVID-19, and on the x-axis is time. So you can see that the blue curve are the people who received the vaccine, and in the yellow curve are the people who received the placebo. So the curves separate on day 7. What this means that as early as 7 days after a single shot of this vaccine, people already have substantial protection. If you click again, then day 28 was the established endpoint of the trial. So that's where the 85% protection comes from day 28. And that's already a stunning result given the harsh epidemiologic conditions in which this vaccine was tested. But I'll actually point out an even more striking result. It's not the primary endpoint of the study, so it can't actually be reported as the endpoint of the study, but just visually look at day 48. After day 48, there was not a single case of severe infection in the vaccinees. And we know the immune responses continue to increase over time. So the endpoint of the trial was 85% protection against severe disease within 28 days. But we know the immune responses continue to mature even after the single shot vaccination. And so these data so far actually show that there was essentially 100% protection against severe disease by day 48. And there were continued accumulation of cases in the placebo group. Next slide. So Galit already covered the issues of the variants of concern and how some of these are more contagious and some of these are resistant to antibodies. The South Africa variant is the one that's generated the most concern because it is partially resistant to antibodies and has been shown to dramatically compromise the efficacy of at least 2 other vaccines that have been tested. So that's why the protection against this virus in South Africa was quite a surprising result, including even for myself, because we would never have expected a vaccine to work this well against this particular virus. It's also not just a South Africa concern, it's actually a concern for the United States because this virus is already circulating in increasing amounts in the United States, and other variants that are essentially analogous, that have the same resistance mutations. You may have heard of the New York variant, for example, or the so-called Brazil variants, which are also circulating in the US. have mutations that are very similar and we imagine would behave analogously to this virus. So for any vaccine to have efficacy in the US and particularly to have efficacy globally, there really needs to be demonstrated protection against the B1351 variant from South Africa. And so far, I think that of the approved vaccines, then the J&J vaccine is the only vaccine that has demonstrated this so far. Next slide. So this shows this in even more clear detail. On the x-axis is time, and on the y-axis is the percent of viruses circulating of different variants. You can see how don't worry about the numbers, but during essentially most of the year, most of last year, there were a variety of different variants that sort of waxed and waned over time without much notice. That didn't, essentially didn't. Not much to note. But then suddenly around November, you see this dramatic emergence of this bright orange virus. And this is the virus of concern. It's actually the B1351, not the 117. So that's an error on the slide. But it's the B1351 variant in South Africa. And the J&J efficacy trial was conducted. The efficacy assessment period was during this block of time shown in red, which was essentially almost completely dominated by this virus. So we know for a fact that the vaccine works against this virus now. Next slide. And just to show what the difference was for the trials conducted last fall, the Pfizer Moderna studies were done at a time of much reduced incidence of disease, and also at a time when there simply was no circulating variants of concern. So there's been a lot of discussion in the press about the difference in the efficacies of these vaccines. And it's really not a comparison that can be made because these trials were essentially done at different points in time in different epidemiologic conditions, and one after the emergence of variants and one before the emergence of variants. But what all the vaccines have in common is that they're safe and they're highly effective, and all the vaccines will prevent the things that people really care about, which is, will I get sick? Will I go to the hospital? Will I die? And all three of the vaccines that are approved in the US will protect you from that. Next slide. Also, this is just to emphasize what I told you before, that the Ad26-based COVID vaccine is a single-shot vaccine, but the antibodies as well as T-cell responses continue to increase over time. And then they actually stay up for a long period of time. We don't know how long for COVID-19 because it's so new, but we know for a Zika virus vaccine based on Ad26, the antibody levels stayed up for at least a year. So we're optimistic, at least, that the immunity raised by this vaccine will continue to improve over time. We often hear about the durability and expect that the antibody titers will drop. But actually, with this vaccine, so far, at least with the 3 months of data we have, the antibody titers actually continue to increase even beyond the day 28 time point. That was the primary endpoint of the trials. Next slide. So there are a lot of different questions. I'm not going to read all these to you, but I think Bruce, Galit, and I would be happy to discuss with you any aspects of either this vaccine or vaccines in general, or really anything about COVID-19 in general that this group would like to talk about. So I'll stop there, and I think there's plenty of time for discussion.
That's great. Galit, Dan, it's terrific to listen to you talk about this and to feel, give people a sense of what it was like right at the center of all the action. And I think we're just so grateful to Terry and Susan and to Mark and Lisa and to others that have supported our program. And we're so delighted to be in this environment here with the academic teaching hospitals, Harvard and MIT, with just such a wealth of talent that enhances the ability of all of us to be able to do the work that we're doing. So I think what we'd like to do now is turn it over to questions. I think some of the questions have already come up. I know Adele Bacow was asking a very important question that probably a lot of people are interested in, and that has to do with, now that people have been vaccinated, what can they do? When can they sort of return to some semblance of normality? And in fact, the CDC released guidelines today that you can find on the CDC website about that. So what I'll do is put those in the chat. But essentially what they're saying now is that if you've been fully vaccinated and you're getting together with people who've also been fully vaccinated, that you can do that without social distancing and without masks. And there are other specifics there as well. So that's really good news. And so I'll put those in the chat for people to be able to look at. Maybe another question that we could ask Dan is, Dan, do these vaccines protect people from getting infected, or do they protect from disease? What's your sense, and what can you say about the J&J vaccine in particular?
So that's a really important distinction. For all of the phase 3 trials done, the endpoints were symptomatic COVID disease. And so up until this point in time, then we've had to always say that we don't really know if any of these vaccines will prevent you from getting infected. All that we know is that they'll prevent you from getting disease, particularly severe disease. But none of the clinical trials done so far have determined whether the vaccines actually prevent infection. And that's important because prevention of asymptomatic infection would then be would then naturally result in decreased transmission, which would allow then people vaccinated to reduce social distancing, as well as greater confidence that vaccinated people won't have an asymptomatic infection and still spread the virus. But what we know is a few things. So first, there is some population data coming out, particularly from the UK and Israel, of the early deployment of the Pfizer vaccine that does suggest an effect, but it's not definitive. And the second piece of data that we have is emerging preliminary data from the J&J phase 3 trial. The fact that it was a 45,000 person trial conducted in the geographies that gave such a surge of infection, the study has tremendous statistical power to ask questions that none of the other studies could do. And so there is a preliminary analysis of the first 3,000 people in the J&J trial that reached the day 71 time point, and antibody analysis, which can differentiate between infection and vaccination. The preliminary analysis has shown that the J&J vaccine gives approximately a 74% reduction in asymptomatic infection, which is, it was de-emphasized in the press releases because it is a preliminary analysis, and we really need to wait until all 45,000 people get to that endpoint and to make it a really powered study. But if that preliminary analysis holds up, then it'll show that at least the J&J vaccine actually prevents infection and not just prevents disease.
Great. And maybe a question for Galit, and I welcome people to write questions into the chat. Galit, what about durability of antibodies from natural infection, and what about immunization for people that have had COVID?
Yeah, two really good questions. So what we know from the very large serosurveillance studies that have come out of Iceland and Europe and now the US as well, we've been tracking antibody responses and T-cell responses in populations now upwards of 9 months from their initial COVID infection. And I think that the data clearly shows that in most individuals, the antibody responses will persist for up to 9 months. And so what that means is that after one hit of infection, you do make antibodies and they will persist. The problem though with COVID infection is that the levels of antibodies that one generates following infection is so variable across populations. Some individuals are infected with COVID and they end up with really high levels of antibodies that will persist over months and months. And some individuals after severe disease will have lower levels of antibodies. And the issue right now is that we don't know the level that is associated with protective immunity. And so this is really where we worry when we think about vaccine deployment across populations, is that we do not want to take it for granted that somebody who has previously seen SARS-CoV-2 and has raised a natural immune response will necessarily be protected. So where the field has moved and where the CDC has made very clear statements and put out guidelines is that even individuals who have previously seen SARS-CoV-2 should be immunized. We want to raise those antibody levels to this very high level where we know that immunity will provide protection from infection. And we want those antibodies to stay as high as possible. Now, while the CDC advocates that individuals who have had COVID should get both vaccines and follow the recommended EUA recommended guidelines for vaccinations, that's one for the prime and boost for Moderna, as well as for Pfizer, or the single shot for J&J. The scientific community is beginning to realize individuals who have been exposed to SARS-CoV-2 may not need a second boost. They may already get a second boost from a first dose of the vaccine. And so I think where we're trying to evaluate the data currently is to try to really understand if that second induction of antibodies will truly get everyone up to those consistent levels that we believe are critical for that highly efficacious result. And so for now, I think everybody stands behind this recommendation that everyone should get both doses of the mRNA vaccines if they have been previously infected.
Great. So here's a question from Helen. Can binding antibodies cause antibody-dependent enhancement? How do we know that creating antibodies to variants will be the safer neutralizing antibodies? Dan, do you want to take that one?
So there's been a lot of discussion, particularly early on during last year, about whether vaccines could sort of backfire, and if they induce the wrong type of antibodies, perhaps non-neutralizing antibodies, perhaps binding antibodies, perhaps low titer antibodies, could it actually do more harm than good? Could it actually make someone more susceptible to infection, or could they make someone more susceptible to having a bad outcome from infection? Clearly, that would put a huge damper on the vaccine plans and would create major safety concerns for vaccines. So that was looked at by all the vaccine developers in various different ways in animal models. And in animal models, there was no evidence that either non-neutralizing, lower titer, sub-protective doses of antibodies cause any sort of worsening of disease or enhancement. But of course, what's important is in clinical trials, and now in very large clinical trials and even larger rollouts, then so far there hasn't been any evidence of enhancement of disease or infection rates by vaccines, even if you specifically look at the breakthrough infections and settings where the vaccines are not successful, then there isn't any evidence with now many millions of people vaccinated of that concern.
Great, thanks very much. So a question from Peter Slavin. Do we know anything yet about impact of the vaccines on the incidence of long haul symptoms? This is a really important issue about the long-term sequelae of infection that has really been debilitating for a number of people that have become infected. And I think is another reason to get immunized. But I'll take a stab at that and then hear from Galit and Dan. My sense is that there really aren't data yet because the vaccines haven't been in use long enough to really generate those data. But if you don't get infected, you're definitely not going to get the long haul symptoms. So since these things are so effective, I think the likelihood of that is not high. Galit, do you want to say?
Yes, there are actually anecdotal cases of that reported this morning at the CROI
Oh,
conference.
great.
Conference for Retrovirus and Opportunistic Infection. So Peter, you're right on. So what was interesting is that, so long haul is kind of broken down into kind of, it's so many different syndromes. And this is the issue with calling it long haul, is that there are so many different ways that COVID can cause long-term consequences. And that is, I think, what we are most scared about, right? So we think about the immediate, as Bruce calls it, existential threat of COVID, right? Which is death. But then there's this bigger class of, I think, threats caused by this virus where people develop, renal disease, cardiac disease, chronic fatigue. Such a crazy spectrum of disease. And so what was interesting is that at least for one class of the long haul, which are people who maintain high levels of viremia, right? So these are people who just can't clear the virus. There were a couple cases reported today at the conference out of Italy that show that if you immunize those individuals, you completely clear the virus and they resolve inflammatory disease. So at least for one segment of the population, it's boosting their response, is truly helping them to ultimately clear this pathogen. So I think that what that means is that there are going to be, situations where the vaccine is going to help, where it's directly against the virus. And there are some cases where we have this long COVID syndrome, which is not really, it was triggered by the virus, but the long-term implications, long-term outcome is really something to do with some reshaping of the immune response. So for those individuals, it may not be helpful, but it's a really positive thought, at least for those individuals who are cooking the virus for long periods of time, where the virus could mutate even further and cause spread of novel variants. So we want to get rid of that compartment or reservoir of virus.
That is terrific news, Galit. And kind of a reflection of just how fast this whole field is moving. I mean, this is hot off the press from this morning, right to your living room. So there's a question here from Robert. Are any of the vaccines likely to protect kidney transplant recipients who are immunosuppressed? Dan, do you want to take a stab at that one?
Sure. So the phase 3 trials, by requirement of the FDA, excludes certain groups of people, including pregnant women, including immunosuppressed rheumatologic patients, organ transplant patients, cancer patients. And that's standard for any development process. And then typically after a vaccine or intervention is shown to work in people without immunosuppressive conditions, then it starts to be used in the so-called special populations, immunosuppressed populations, pediatric populations, pregnant women, et cetera. For COVID-19, everything worked so quickly that essentially as soon as there was an interim analysis, not even a final analysis, and actually none of these COVID trials have presented their final analysis yet, but based on just the interim analysis, then the vaccines are approved for emergency use and rolled out to essentially the entire country, and in some cases the world. So we're in a situation where currently there's really no information, there's really no data on these special populations, but there is clinical data that's being accumulated, and in most cases the professional societies are recommending immunization of these populations regardless. There's little reason to be concerned about safety concerns, although formally they haven't been tried, because the RNA and the vectors, they don't involve any replicating virus components. So they wouldn't, there's no reason to suspect they would be dangerous in those populations. What's just not known is whether people will respond to the vaccines in the same way, or whether people who have immunosuppressive conditions will have a reduced antibody response. But a lot of that data is going to be determined essentially just from the clinical rollout of the vaccines. So the short answer is we don't know yet about those populations, but there will be a lot of data that emerges over the coming months.
And Dan, maybe to balance that, what would your concern be for somebody who's immune compromised with a kidney transplant if they get COVID disease?
Right. So the balance is that people who are immunosuppressed, who can't mount their own immune responses as well as a normal healthy person, likely would have a much more serious course following COVID disease. So it's even more important to protect them from the disease. So that balance, even though there's not formal data in those populations, that thinking and that rationale has led most of the professional societies and physicians to vaccinate essentially all those populations, except for kids, because for kids, we actually don't know what dose to use. And we don't want to give kids a dose that is too reactogenic for them. So for kids, there will have to be clinical trials.
So here's one, maybe this is for you as well, Galit or Dan. How long will this is from Lori Slavin, how long will it take to produce and distribute the next generation vaccines Galit mentioned to target the new variants?
That's a Dan question.
Okay, Dan, you're on.
I believe that the Moderna study with the mRNA expressing the B.1.351 variant, that trial is going to start soon.
I think it's actually started, hasn't it? At least it's been delivered down to the NIH.
Yeah. So if it hasn't started already, it'll start soon. But I think, and all the other companies will follow suit. But the question is, how would you actually interweave that into a national and a global rollout program that's already being done at scale? It would be incredibly difficult to call everyone back for a booster. It could be done if needed. It's certainly possible, but it would be difficult and confusing to people. And then do you eventually, in the middle of a rollout, do you stop giving the current vaccine and give them a combination of the two vaccines, for example, and then call back the people who received the vaccine earlier? Anything is possible if it's needed, but it will make the rollout more complicated for sure. And if that is required, it will delay the time period before everyone is so-called fully vaccinated. So really the question is that going to be needed? And that's not yet known. So I think that the companies are doing the right thing in terms of preparing vaccine constructs, expressing the immunogens of the variants. So they will be there if needed. Whether they'll be needed, and if so, how they will be rolled out on a global scale is not yet known, but presumably will be dependent largely on population effects. And basically, if there's breakthrough in populations on a population level, then I think the revised vaccines will be deployed. It would be pretty difficult now to essentially change the entire plans of the rollout.
So it's a few minutes past 6. I want people to feel free to leave if they need to. We said we'd go to 6, but there are a few more questions, so we'll just answer those in the next few minutes, and then we'll end. There's a question from Mark Schwartz. What's the likelihood the disease becomes endemic and requires annual shots? Which of you wants to answer that? There we go, Galit. I saw you unmuted.
Yeah, so I think Mark, the epidemiologists have modeled, what level of protection or efficacy would we need in order to completely get rid of the virus. And I think that level of transmission blocking efficacy is not really clearly induced by the current vaccines that we have in our toolkits. The data that Dan was referring to out of the UK and Israel suggests about 60% transmission blocking activity, but against a respiratory pathogen, if you can't get every single person immunized in a population, it doesn't seem that we're going to completely obliterate this pathogen from the face of the earth. So what it looks like is that this virus will continue to spread until we can get, upwards of, I think they've estimated 80% coverage in the population. And even then, most likely it will end up becoming a circulating pathogen. I think the question becomes, will this become an annual vaccine, right? I think that's what everyone wants to know. Or is it going to be a once in a lifetime vaccine that we get as kids, right? Once we get enough of the population covered. And I think that, what we're seeing with the variants, as Dan just mentioned, is if we can cover the variants and anticipate any new emerging variants of concern that we can cover with these vaccines, then it might end up moving towards a once in a lifetime where we cover the population as well as we can in this fall swoop of global deployment of vaccination. get the kids vaccinated, because that's where we're going to get herd immunity. Without getting kids vaccinated, we're never going to reach herd immunity. And that's one of the most important points that I think that we have to stress here. And then once we have enough of the population covered and we can reduce the death and burden of disease, right, that's overwhelming hospitals, then it might become an annual. If variants pop up that are able to escape and still cause devastating disease, then we may end up moving to an annual, which would not be different, right, than the flu vaccine. And that would be okay, I think.
Yeah, great. Question from my sister, Lynn Walker. What are the plans for vaccinating children? I mean, I think we started to talk about that, but what do you think realistically, Dan? I mean, I think of all the vaccines, the J&J vaccine, your Ad26 platform has been tested down to very young children. Do you want to comment?
Sure. So the challenges for pediatrics is that in general, the FDA does not allow products to be tested in kids until they've been proven to be safe and effective in adults. Now, 3 vaccines have been proven to be safe and effective in adults. And I think all 3 of the companies, namely Moderna, Pfizer, and J&J, as well as AstraZeneca abroad, have started pediatric studies. The other requirement from the FDA is that there has to be an age de-escalation. So trials have to start in teenagers, then go down to younger kids, and then at the very, very end, babies. And that normally is a slow process, because normally there needs to be safety, at least safety and immunogenicity shown in each group before you can go down to a younger and younger age group. So it does take time. But I think all the companies are very aware that it would be hugely advantageous on many different levels if we could get the entire school-age population of kids vaccinated by the time they have to go back to school in September. Not just to protect their own health, but also to allow the workforce to resume, so parents don't have to stay home with their kids. They can go back to work for all the reasons, not just medical, but societal, social, etc. So I do think that it is going to be a very high priority. And we know that under the current administration, opening up schools is one of the absolute top priorities. So I think there's going to be a big emphasis on trying to accelerate the pediatric program. And I think the companies are willing and able to do that. And if the regulatory frameworks are in place, then it can and should be done. The Ad26 J&J vaccine is particularly well suited for that, because it has already been age de-escalated, not for COVID, but for an RSV vaccine, respiratory syncytial virus vaccine, in which the Ad26 RSV vaccine from J&J was age de-escalated down to babies 4 months of age. So although an Ad26 COVID vaccine would also need to undergo the same age de-escalation, it likely can go faster because there is already demonstration of the vaccine platform being safe and immunogenic in kids all the way down to young babies. So it's actually something that I'm actually talking with them about, and I'm personally encouraging my colleagues at J&J to accelerate the pediatric program.
That's terrific. So there's a question from David Gagnon, and then we're getting some second questions from people. I'm happy to arrange for answers to those, but I think we'll skip those now just in the interest of time and go to David's question. What are your thoughts on which vaccine might be safer for individuals with severe allergies? Galit.
No, I'm giving that one to Dan.
Oh, you're giving that one to Dan.
Generously handing it to Dan.
Okay, Dan.
Well, these are all new. So rare side effects often are not apparent in clinical trials, even large clinical trials of 30 or 45,000 people. For example, the anaphylactic reactions seen with the Pfizer and Moderna vaccines, I believe they're believed to occur in about 1 in a few 100,000 individuals, which is a very rare side effect, but it is a life-threatening reaction. Thankfully, it's very easily diagnosed, very easily treated. But that was not the concerns of severe allergic reactions like anaphylaxis were not apparent until the 1 million dose rollouts. Because I mean, there's no way to detect an adverse event in, say, 1 in 100,000 or 1 in a million people in a clinical trial of 30,000 people. So, there is a rare incidence of severe allergies, including anaphylaxis, to the RNA vaccines. It has been hypothesized that is due to an allergy to a component, an ingredient in the RNA vaccine called polyethylene glycol, which is actually part of many other medicinal products and is a well-known clinical entity. It's well known even prior to these vaccines that some people have allergies to polyethylene glycol. That's not proven, but that's been, I think, the lead contender for the source of the allergic reactions to the Pfizer and Moderna vaccines. The J&J vaccine, I would actually say it's too early to tell because it just has started the 1 million dose rollout the last week. I didn't hear of any anaphylactic reactions last week, which is good. The anaphylactic reactions were seen with the Pfizer vaccine, I think, within 2 days of the rollout in the UK and then the US. But I think it's too early to really answer your question. So I can't say for sure that the J&J vaccine won't trigger severe allergies or anaphylactic reactions once it's given to 10 million, 100 million people. We won't know that until it's done. What I can say is that it won't be a problem for people with polyethylene glycol allergies because it doesn't contain polyethylene glycol. So whether people will have allergic reactions to other components of it is yet to be seen, I think. So if you have a polyethylene glycol allergy, which is known but exceedingly rare, then you're probably better off not getting a vaccine containing that ingredient. Other than that, I think we can be assured that all of these vaccines are very safe. And if there is that life-threatening severe allergy, then it's detected often within minutes, and there's a very effective medication for it that's given sort of on site at any of these vaccination centers.
Great. Well, I want to thank Dan and Galit for spending time going through this and for their terrific presentations and their terrific insights and information that they've conveyed. I want to thank Larry Bacow and Peter Slavin and Raphael Wright for supporting the Ragon Institute and allowing us to operate in this phenomenal environment. You know, what Terry and Susan have allowed us to create here is really something that has been transformative for all of us. I want to thank Mark Schwartz for getting involved very early in this whole process and supporting Dan in the development of the vaccine. And I want to thank all of you for participating. It's been a pleasure, and we'll do another one of these updates in the future. So thanks very much.
Thanks.
And thank you, everybody, for joining. I hope you enjoyed and learned something. And if there's more questions, we'll get some answers to people so that everybody's voice is heard and questions are answered. And it's really great to see everyone. So I can't wait till we're all vaccinated and we can hang out again.
Oh, and I see that Larry Bacow just wrote, asking, who's the president of Harvard, asked, can I say a word at the end? Larry, you most certainly can.
Thank you very much, Bruce. I just wanted to say, and I think I speak not just for myself, but also for Raphael. And Peter's on this call. Peter can speak for himself, but we're so privileged to be able to partner with all of you. And thanks to Terry and Susan for having the vision to create the Ragon, and so many of you for having supported it. But I also just wanted to note that Bruce and Galit and Dan are not only spectacular scientists, you guys are also great teachers. And so thank you for teaching all of us so well. And it's a privilege to be a student in your classroom.
Well, on that note, thanks everybody. We'll sleep well tonight.
Okay. Good night, everyone. Thanks for joining.
All right.
Stay safe.
Thank you.
Okay.